Good morning, class. So today we will discuss lymphatic system and the immunity. So to start with, the lymphatic system is very important for the protection of our body. So what are the functions of the lymphatic system? So it function for fluid balance, lipid absorption, and defense. So for fluid balance, the excess interstitial fluid enters the lymphatic capillaries and becomes lymph. For lipid absorption, it absorbs fat and other substances from the digestive tract and provides defense against microorganisms and other foreign substances by filtering the microorganisms from lymph by lymph nodes and from blood by spleen. So what are the anatomy of the lymphatic system? So if we could see the picture, so we have different organs of lymphatic system. We have the tonsils, the thymus, the lymph nodes, the spleen. Yeah. So what are lymphatic capillaries? So they carry lymph away from tissues. So lymphatic capillaries are present in most tissues of the body, except in the central nervous system, bone marrow, epidermis, and cartilage. It is more permeable than blood capillaries, and the epithelium functions as a series of one-way valves. So it also collects excess interstitial fluids from the dermis and subcutaneous tissue, and a deep group collects excess fluids from the muscle, viscera, and other deep structure. While our lymphatic vessels, so they are lymphatic capillaries that join together to form the lymphatic vessels. So they have bulbs that ensures one-way flow and resemble small veins. So the compression of the lymphatic vessels causes the lymph to move forward through them. So there are three factors that compress the lymphatic vessels. So the first one is the contraction of the surrounding skeletal muscle during its activity, periodic contraction of the smooth muscle in the lymphatic vessel, and lastly, the pressure change in the thorax during breathing. So the lymphatic vessels converge and eventually empty into the blood at two locations in the body, which is the right lymphatic duct and the thoracic duct. So what is lymphatic tissue? So lymphatic tissue is composed mainly of lymphocytes. It can be encapsulated or non-encapsulated, and it has fine reticular fibers that holds lymphocytes and other cells in place and traps microorganisms and other items in the fluid. While our lymphatic nodules are numerous in loose connective tissue of digestive, respiratory, urinary, and reproductive system. While our tonsils are a large group of lymphatic nodules found in the nasopharynx and the oral cavity. It provides protection against bacteria and other harmful material. So there are three groups of tonsils, the palatine, pharyngeal, and the lingual. Among these three, the palatine is pair. Yan. So we have two palatine tonsils that are located on each side of the posterior opening of the oral cavity. We have the pharyngeal, located near the internal opening of the nasal cavity, and the lingual, or located on the posterior surface of our tongue. So the adenoids are happens when the pharyngeal tonsil is enlarged and can interfere with normal breathing. So the removal of the palatine tonsil is adenoidectomy, and the removal of the pharyngeal tonsils is tonsillectomy. So what are lymph nodes? So rounded structures varying from the size of small seed to that of a shelled almond. So distributed along the various lymphatic vessels, and most lymph passes through at least one lymph node before entering the blood. So it can be found throughout the body and classified as superficial or deep. So there are three superficial lymph nodes on each side of the body. So they are found in the inguinal nodes in the groin, the axillary nodes in the axilla or armpit, and the cervical nodes in the neck. So kung napapansin nyo kapag nagpapacheck up kayo, isa sa mga ginagawa ng ating mga doctors is to check the lymph nodes in our cervical or in our neck part. So kinakapa nila kung namamaga ba yung ating lymph nodes. So they will know if there's an infection ongoing. So the lymph enters the lymph node through afferent vessels and passes through the lymphatic tissue and exits through efferent vessels. So when we say afferent, going to the body, and kapag efferent, it means away from the body. So activate the immune system and stimulate lymphocytes in the lymphatic tissue to divide and removes pathogens from the lymph through the action of macrophages or through phagocytosis. While our next organ is the spleen. So roughly the size of a clenched fist and is located in the left superior corner of the abdominal cavity. It has two specialized types of lymphatic tissue, the red pulp and the white pulp. So the white pulp is a lymphatic tissue surrounding the arteries within the spleen, while the red pulp is associated with the vein. So the main function of the spleen is to filter blood instead of lymph. So the cells within the spleen detect and respond to foreign substances in the blood and destroy old and damaged RBC. So another function of the spleen is to become the blood reservoir. So the spleen can be ruptured in a traumatic abdominal injury that can result in severe bleeding, shock, and death. So surgical intervention may stop the bleeding. And if there is cracks in the spleen, it can be repaired using sutures and blood clotting agent. So a mesh wrapped around the spleen can hold it together. So the removal of spleen is called the splenectomy. Then we have another organ, which is the thymus. It is a bilobe gland, roughly triangular in shape and located in the superior mediastinum. So between the two lungs and inferior to the heart. So the partition dividing the thoracic cavity into left and right parts. So it has two divisions, the cortex and the medulla. So when we say cortex, the lymphocytes are numerous and form dark staining areas, while the medulla has fewer lymphocytes with a lighter staining and found in the central portion of the lobules. So thymus is the site for the maturation of T cells. So large number of T cells are produced in the thymus, but most degenerate. So the T cells that survive the moderation process are capable of reacting to pathogens. So our T cells help protect against pathogens, and the production of T cells declines later in life due to decreased thymus function. So kapag tumanda na or adult na ang isang person, the thymus shrinks and will be replaced by fat. So, hindi mo na siya makikita. So, lumiliit yung ating thymus once we age. So, next natin is immunity. So, the immunity is the ability to resist damage from microorganisms, harmful chemicals, and internal threats such as cancer cells. So there are two types of immunity, innate and adaptive immunity. So when we say innate, it is non-specific resistance to pathogens. So it prevents entry or remove microbes, but the response to them is the same each time the body is exposed. So there are chemical mediators that are involved that promotes phagocytosis and inflammation, as well as cells that also involve in phagocytosis and productions of chemicals that are being released to prevent or remove the microbes. Then we have the adaptive or specific immunity, wherein the body recognizes and destroys the pathogen, but the response improves each time the pathogen is encountered. So during adaptive immunity, it is specific and it has a memory. So specificity is the ability to recognize a particular substance, and memory is the ability to remember previous encounters with a particular substance and respond rapidly. So the more na encounter siya ng body, the more na nakikilala siya ng katawan, and mas alam na ng body and mas mabilis na yung magiging reaction afterward. Yan. So this is the difference between innate and adaptive immune system. So in our innate immunity or rapid response, we have the macrophage, the mast cells, the natural killer cells, the complement proteins, the dendritic cells, neutrophils, basophils, and the eosinophils. While in our adaptive immunity, we have the B cells, antibodies, the cytokines, and the T cells that are differentiated into CD4 and CD8. So what are the innate immunity? So they are the physical barrier. So it prevents pathogens and chemicals from entering the body in two ways. So the skin and mucous membranes are the one that form barriers that prevent their entry, as well as the tears, saliva, and urine that washes substances from the body surface. So the pathogens cannot cause a disease if they cannot get into the body. Then we also have chemical mediators. So they are molecules responsible for many aspects of innate immunity. So some chemicals on the surface of the cells destroy pathogens or prevents the entry of some microorganisms. So during this time, they promote inflammation that causes vasodilation, increase vascular permeability, and stimulates phagocytosis. So there are two ways. We have the complement system and by the stimulation of the interferon. So during the complement pathway or complement proteins, they are a group of more than 20 proteins found in the plasma that circulates in the blood in inactive form. It can be activated in cascade form by classical or alternative pathway, while our interferons are proteins that protect the body against viral infection. So what they do is that they bind to the surface of the neighboring cells where they stimulate those cells to produce antiviral protein. So some interferons play a role in activating the immune cells such as the macrophages and our natural killer cells. Yan. So this is the complement cascade. So a while ago, I said that there are two ways, there are two pathways, the classical pathway and the alternative pathway. So the start of the classical pathway is when the antibody has been bound to the antigen. So at this point, the C1 will be activated, while our alternative pathway is activated when the complement C3 becomes spontaneously active and combines with foreign substances and factors B, D, and E. So complement protein C3 to C7 promotes phagocytosis and inflammation as well as chemotaxis. So while our complement protein C5 to C9 combine to form a hole in the plasma membrane of the target cell causes the cell to lyse. Then we have our white blood cells. So our white blood cells are a part of our innate immunity. So they are cells derived from them are the most important cellular components of immunity. Diba, katulad nung nabanggit ko sa last meeting, yung ating neutrophil, so they are usually the first line of defense and usually they are elevated if there is a bacterial infection while our lymphocytes increases if there is a viral infection and are basophil and eosinophil if there are allergic reaction and possible worm infection. Yan. And our monocyte acts as a phagocyte. Yan. So they are produced in the red bone marrow and lymphatic tissue and released in the blood. So important chemical known to attract WBCs are the leukotrienes, kinins, and the histamine. So the movement of the WBCs toward this chemical is called the chemotaxis. So phagocytic cells. So phagocytes, cells that protect the body by ingesting harmful foreign particles, bacteria, and dead or dying cells. So phagocytosis is the process of ingestion and destruction of particles by phagocytes. So example of WBCs are the neutrophils and our macrophage. Then the cells of inflammation, we have the basophil, which appear in many specific kinds of inflammatory reactions that particularly that causes allergy symptoms. So it contains anticoagulant heparin, which prevents blood from clotting too quickly. And they also contain a histamine that promotes blood flow to the tissues. Then we also have the mast cells, which is best known for their role in allergy and anaphylaxis. So mast cells play an important protective role as well, being intimately involved in wound healing. Angiogenesis, immune tolerance, defense against pathogens, and the blood-brain barrier function. Then we also have the eosinophils that participate in inflammation associated with allergies and parasitic infection as well as asthma. Then we have our natural killer cells. So they are type of lymphocytes that are produced in the red bone marrow and recognize classes of cells such as tumor cells or virus infected cells. So they release chemicals that damage cell membrane and cause the cells to lyse. Then we have inflammatory response. So tissue injury, regardless of type, can cause inflammation. So when initiated by chemical mediator, the response will be vasodilation, chemotactic attraction, and increased vascular permeability. So there are two types of inflammatory response. We have the local and the systemic. So the local involves redness, heat, swelling, pain, loss of function in the inflammation, while the systemic inflammatory response are increase in neutrophil numbers, fever, and possible shock. Yan. So when the bacteria enter the tissue will be damaged, and that will cause uh, the release of the chemical mediators and increases blood flow and increase the vascular permeability. This will also stimulate chemotaxis and will increase the number of the WBC and chemical mediators at the site of tissue damage. So once the bacteria are contained, destroyed, and phagocytized, the bacteria will be removed from the body and the tissue repair will start. But if the bacteria remains after phagocytosing, uh, additional chemical mediators will be activated. Then next we have adaptive immunity. So it involves the ability to recognize, respond to, and remember a particular substance. So it has stimulants, so the antigens and the haptens. So antigens are large molecules. So there are two types, the foreign antigens and the self-antigens. So the foreign antigens are not produced by the body and they are usually introduced from the outside. Then we have the self-antigens that are produced by the body. While haptens are small molecules incapable of combining. So there are two types of adaptive immunity. We have the humoral immunity and cell-mediated immunity. So when we say humoral, it is the B cells that acts upon the immunity, while in cell-mediated, it is acted upon by our T cells. So B and T cells are originated in the red bone marrow and move to the lymphatic tissue from the processing site and continually circulate. So clones are small groups of identical lymphocytes. So kanina na nabanggit ko, so si T cells will mature in our thymus. So origin development of the lymphocytes. So positive selection. So it ensures survival of the lymphocyte that react against the antigens, while negative selection is the elimination of the lymphocytes that react against self-antigens. So the primary lymphatic organs includes the red bone marrow and the thymus, where the lymphocytes mature into functional cells. And we also have the secondary lymphatic organs where lymphocyte produces an immune response. So what are antigenic determinants? So determinants are specific regions of a given antigen recognized by a lymphocyte, while our antigenic receptors are the surface of the lymphocyte that combines with the antigenic determinant. So major histocompatibility complex or MHC. The most lymphocyte activation involves glycoproteins and of cell surfaces called the MHC molecules. So there are two types of molecules, the class 1 and the class 2 molecules. So for the class 1, it displays antigens on the surface of the nucleated cells resulting in the destructions of cells, while class 2 display antigens on surface of the antigen-presenting cells, resulting in activation of the immune cells. Co-stimulation. So it can be achieved by cytokines, which are proteins or peptides secreted by one cell as a regulator of neighboring cell. Then we also have the proliferation of helper T cells. So after the antigen is processed and presented to a helper T cell by a macrophage, the helper T cell responds by producing interleukin-2 and interleukin-2 receptors. So this IL-2 binds to the receptors and stimulates the helper T cells to divide. And the daughter helper T cells produced by this division can again presented with antigen by macrophages and again be stimulated to divide, thus increasing the number of our helper T cells. Next is proliferation of the B cells. So before a B cell can be activated by a helper T cell, the B cell must be phagocytized and process the same antigen that activated the helper T cell. The B cell uses MHC class 2 molecule to present the process antigen to the helper T cell, and the T cell receptor binds to the MHC class 2 antigen complex. Then there will be co-stimulation between the B cell by the CD4 and other surface molecule, and there will also be stimulation by IL released from the helper T cell. So the B cell will divide, resulting into daughter cells, and will divide more to produce many cells. So many of the daughter cells differentiate to become the plasma cells, which produces our antibodies. So the antibodies are part of the immune response that eliminates the antigen. So, laging tatandaan na si plasma cell, siya ang nagpo-produce ng ating antibodies. So, lymphocyte inhibition. So, tolerance. So, to prevent the immune system from responding to self-antigen, it is provoked by deletion of self-reactive lymphocytes, prevents activation of lymphocyte, and activation of suppressor T cells. So, what is antibody-mediated immunity? So, this is the first type of adaptive immunity. So, this uses antibodies or immunoglobulins. So, they are make up a large portion of proteins in the plasma and can be separated into albumin and alpha, beta, and gamma globulin portions. So antibodies are sometimes called gamma globulins because they are found mostly in the gamma globulin part of the plasma. So there are 5 types of immunoglobulin: the IgG, IgM, IgA, IgE, and IgD. So the variable region of the Ig, the immunoglobulin, is the part that combines with the antigenic determinant of our antigen. Yan. So among these 5, the most highest molecular weight will be our IgM because it contains more branches compared to the other immunoglobulins. But among the 5, the highest amount that can be found in the body is our IgG. But the first one to respond is our IgM. While our IgE responds during allergic and parasitic infection, while IgA are secreted in the mucus, tears, and saliva, while IgD acts as a B cell receptor. So among these five, only the IgG and IgM access the complement system. So what are the effects of antibodies? So it has two effects. It can be direct or indirect. So for direct effects, it occurs when a single antibody binds to an antigen and inactivates the body. The antigen, or when many antigens are bound together and are inactivated by many antibodies. So example nito is the blood typing, while our indirect is most of the effectiveness of the antibody. So after an antibody has attached by its variable region to an antigen, the constant region of the antibody can activate other mechanisms that destroy the antigen. And so example natin is blood typing. So for our group A, we have anti-B antibodies in the group A, anti-A in the group B, none in group AB, and both for group O. Then we don't have any antigens in the group O and has two antigens for group AB and so forth. And antibody production. So the production of the antibodies after the first exposure to an antigen is different from the following a second or subsequent exposure. So during primary response or first exposure, when the antigen binds to the antigen binding receptor in the B cell, has been activated by a helper T cell. The B cell undergoes several divisions to form plasma cells and memory B cells. So the plasma cell, like what I've said earlier, produces the antibodies. So it takes normally 3 to 14 days to produce enough antibodies to be effective against the antigen. So primary response causes symptoms, while our secondary response or memory response, the memory B cells are the ones that are responsible. So it occurs when the immune system is exposed to an antigen against which is already produced a primary response. So it provides better protection than primary response, and the time required to start producing antibodies are less. And there will be more plasma cell and antibodies produced, and the antigen can be quickly destroyed and no disease symptoms, and the person will become immune. Also, it includes the formation of new memory cells that provide protection against additional exposures to a specific antigen. So during first exposure, the primary response, it has longer response or longer, it takes longer time to produce plasma cell. While in our secondary exposure or secondary response, it has shorter response time and produces more memory B cells and more plasma cells, so it means more antibodies. Then our second type of adaptive immunity is cell-mediated immunity. So as a function of cytotoxic T cells and is most effective against microorganisms that live inside the body. It is essential for fighting viral infections and also involved with some allergic reactions, control of tumors, and graft rejection. So antigen activates effector T cells and produces memory T cells. And these cytotoxic cells are responsible for the immediate cell-mediated immune response, and the memory T cells provide a secondary response and long-lasting immunity in the same fashion as memory B cells. So the cytotoxic T cells lyses virus-infected cells, tumor, and tissue transplant, and produces cytokines which promotes phagocytosis and inflammation. So what is acquired immunity? So there are four types of acquired immunity. We have the active natural immunity, the active artificial immunity, the passive natural immunity, and the passive artificial immunity. So when we say active natural immunity, result from the natural exposure to an antigen, such as disease-causing microorganism that stimulates the immune system to respond against the antigen. While the active artificial immunity, it is in the form of vaccination or vaccine. So it results from deliberate exposure to an antigen introduced to an individual to stimulate the immune system. So usually it is a killed microorganism or weakened microorganism that are being injected to your body to produce a immune response. Then we also have passive natural immunity, wherein it is the result of transferring an antibody from the mother to her baby. And we have the passive artificial immunity, wherein it is result from the transfer of antibodies from an immune animal to a non-immune one. Example of this is the anti-serum. Yan. So, natural active immunity. So, if a person develops an infection, then artificial through vaccination, then natural passive immunity. So, from mother to baby. So, usually through breast milk. And we have the passive artificial immunity. They are the antibodies received from a medicine, from a gamma globulin injection or infusion. So, what is immunotherapy? So, treats disease by altering immune system function or by directly attacking harmful cells. So, the tumor cells bind to the T cells to deactivate them. Thus, the immunotherapy drugs can block tumor cells from deactivating the T cells. So, yun ang ano nila, tinutulungan ng immunotherapy drugs na ma-block yung tumor cells from deactivating the T cells. So what are the types of immunotherapies? We have antibodies, the checkpoint inhibitors. It can also be done by giving vaccine and by giving or transfusing T cells. So antibodies, so a version of protein that can attach themselves to cancer cells, tagging them for immune destruction. And the checkpoint inhibitors, so some proteins help control the immune system, so wherein they can turn off the immune cells that are able to kill cancer cells better. And vaccines, then for T cell transfers, upgrade and improve a patient's T cell. So what are the conditions or disease of the lymphatic system and immunity? So we have lymphedema and lymphoma. And for our immune system, we have the immediate allergic reactions such as the asthma and anaphylaxis. And for delayed allergic reactions, we have the immunodeficiencies such as the SCID and our AIDS. So what are the effects of aging? So little effect on lymphatic system. So there will be a decreased ability of helper T cells to proliferate in response to antigens, so decreased primary and secondary antibody response, and decreased ability of cell-mediated immunity to resist intracellular pathogens. So kapag tumatanda na po, mas mahina na yung immune system. So they are more prone to infection. So it's very important that when you reach the old age, so you have to be very careful and um, be very mindful of your health because your immune system is not that strong as you are when you are young. So that's the end of our lecture. Thank you so much for listening and I hope you learned something today. So have a good day.